Humans have two alleles for each gene. This is because gametes contain one allele for a particular gene. So when two gametes fuse, they produce the genotype, the specific genetic constitution of chromosomes for the offspring. Each pair of alleles can be heterozygous or homozygous. Each allele can either be dominant or recessive. In rare cases, alleles can be double recessive. Genetic diagrams can be used to predict the phenotypes or genotypes of offspring from the alleles of the parents. Monohybrid cross. Monohybrid inheritance is when the inheritance of a characteristic is controlled by a single gene. The diagram below shows how wing length is inherited in fruit flies. Codominance is when both alleles are expressed in a phenotype, neither one is recessive. One example is sickle cell anemia in humans. The phenotypic ratio of this genetic diagram is 1 to 2 to 1. Dihybrid crosses help you work out the chances of offspring inheriting different combinations of characteristics. They show how two different genes are inherited at the same time. The diagram below shows how the texture and color of pea seeds is inherited. Dihybrid crosses work with codominant alleles, but the phenotypic ratio produced is different. Sex linkage. Males are more likely to inherit sex-linked disorders such as color blindness or hemophilia, or fragile X chromosome. Genes that are on the same autosome are said to be linked because they will stay together during independent segregation, so are likely to be inherited together. The only reason this may not happen is if the alleles are split up during crossing over. The closer together the two alleles are on the autosome, the more close they are, the more likely they are to be inherited together. This is because they are less likely to be separated during crossing over. An epistatic gene is one which masks the effect of another gene. Epistatic genes can have dominant or recessive alleles. You need two recessive alleles in order to mask another gene. You only need at least one dominant allele in order to mask another gene. Mutations are any change to the base nucleotide sequence of DNA. If there is a change to the base sequence, the primary structure of the order of amino acids will be changed. This means the location of the ionic bonds in the tertiary structure will be different, so the protein will have a different 3D shape. If this protein is an enzyme, it could mean that the active sites have now lost their specific shape, so can no longer form enzyme-substrate complexes. If this protein is an enzyme, it could mean that the active site has now lost its specific shape, so can no longer form enzyme-substrate complexes. Some mutations can result in genetic disorders or increase the risk of developing certain diseases. Addition mutations. This is when one or more extra bases are added into the base sequence. Changing the number of bases in the sequence results in a frame shift. These mutations often result in a different or non-functioning protein being coded for. Deletion mutations. This is when one or more of the bases are deleted from the base sequence. This, like addition mutations, also changes the number of bases in the sequence, so it results in a frame shift. This, like addition mutations, also changes the number of bases in the sequence, so results in a frame shift. Substitution mutations. This is where a base is swapped for a different one. Substitution mutations do not cause a frame shift. one of the amino acids in the sequence is changed, which still causes a slightly different protein to be produced. Mutagens are factors that increase the rate of mutations. Mutations occur spontaneously when the DNA code is misread, but things like UV radiation, some chemicals, and some viruses increase the probability of this happening. Cancer can be caused by acquired mutations, tumor suppressor genes, and proto-oncogenes. If a mutation occurs in a tumor suppressor gene, the protein that stops cell dividing doesn't function and the cell divides uncontrollably. This can form an immortal clone. If a mutation occurs in a proto-oncogene, the gene can become overactive, producing too much of the protein, causing uncontrolled cell division. A mutated proto-oncogene is called an oncogene. Malignant tumor. Malignant tumors are cancers that divide rapidly and invade and destroy surrounding tissue. Cells can break off and spread to other parts of the body. This is called metastasis. Benign tumors are not cancerous tumors. They grow slowly and are covered in fibrous tissue, which stops them from invading other tissues. Although mostly harmless, they can cause blockages and put pressure on other tissues. Stem cells are undifferentiated cells that can specialize into other types of cells. There are different types of stem cells. Totipotent. Totipotent stem cells can differentiate into any type of body cell. These cells are only present in mammals for the first few cell divisions of an embryo. Pluripotent stem cells. Pluripotent stem cells are found in embryos after the first few cell divisions. They can differentiate into any body cell, but they cannot differentiate into the cells that make up the placenta. Multipotent stem cells. Multipotent stem cells can differentiate into a few different types of cells. For example, stem cells found in adult bone marrow can differentiate into red and white blood cells. Unipotent. Unipotent stem cells can only differentiate into one cell type. For example, there is a type of stem cell that only differentiates into epidermal skin cells. Only specific genes are transcribed and translated so that particular proteins are produced so the cell can carry out its specific functions. Once these changes have been made to a cell, it cannot be reversed to become a stem cell again. One mutational event during development can cause many of the initial stem cells to carry and transmit that mutation. By controlling how genes are transcribed and translated, we can regulate which genes are expressed. All cells contain all the same DNA, but not all the genes are expressed in every type of cell. When different genes are expressed, different proteins are produced, which means the cell has a different structure and therefore carries out different functions. Transcription factors. In eukaryotes, transcription factors move from the cytoplasm into the nucleus through the nuclear pores. In the nucleus, they bind to the DNA near the start of their target gene. They control expression by affecting the rate of transcription. Some transcription factors, called activators, encourage the binding of RNA polymerase to the DNA. This increases the rate of transcription. Some transcription factors, called activators, encourage the binding of RNA polymerase to the DNA. This increases the rate of transcription. Other transcription factors, called repressors, block RNA polymerase from binding to the DNA. This decreases the rate of transcription. Estrogen is a steroid hormone that can bind to a transcription factor called an estrogen receptor. When this happens, the transcription factor becomes activated and is able to bind to DNA in the nucleus. This picture provides a strong overview of the nature of transcription factors.